Dear viewers, this video is part 4 in the 4-part series on cancer cell biology in the Medicine for Anyone YouTube channel. Apoptosis and other mechanisms of cell death. Tissue homeostasis requires a balance between the death of aged, terminally differentiated cells or severely damaged cells and their renewal by proliferation of parent cells. Genetic damage to growth regulating genes of stem cells could lead to catastrophic results for the host as a whole. Thus, genetic events causing activation of oncogenes or loss of tumor suppressors, which would be predicted to lead to unregulated cell proliferation unless corrected, usually activate signal transduction pathways that block aberrant cell proliferation. These pathways can lead to a form of programmed cell death, that is apoptosis, or irreversible growth arrest, that is senescence. Much as a panoply of intra and extracellular signals impinge upon the core cell cycle machinery to regulate cell division, so too these signals are transmitted to a core enzymatic machinery that regulates cell death and survival. Cells, including cancer cells, can also undergo other mechanisms of cell death, including autophagy, that is, degradation of proteins and organelles by lysosomal proteases, and necrosis, digestion of cellular components and rupturing of the cell membrane. Necrosis usually occurs in response to external forces, resulting in release of cellular components, which leads to inflammation and damage to surrounding tissues. Although necrosis was thought to be unprogrammed, evidence now suggests that at least some aspects may also be programmed. The exact role of necrosis in cancer cell death in various settings is still being determined. In addition to its role in cell death, autophagy can also serve as a homeostatic mechanism to promote survival for the cell by recycling cellular components to provide necessary energy. The mechanisms that control the balance between enhancing survival versus leading to cell death are still not fully understood. So, dear viewers, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Metastasis. The metastatic process accounts for the vast majority of deaths from solid tumors, and therefore an understanding of this process is critical for improvements in survival from cancer. The biology of metastasis is complex and requires multiple steps. The initial step involves cell migration and invasion through the extracellular matrix. Note, the extracellular matrix is a non-cellular meshwork of cross-linked macromolecules including collagens, proteoglycans, and glycoproteins that form a dynamic supramolecular scaffold. It provides cues, both physical and chemical, which influence cancer progression and metastasis. The three major features of tissue invasion are: 1. Cell adhesion to the basement membrane, 2. Local proteolysis of the membrane, and 3. Movement of the cell through the rent in the membrane and the ECM, that is extracellular matrix. Cells that lose contact with ECM normally undergo programmed cell death, and this process has to be suppressed in cells that metastasize. Another process important for many metastasizing epithelial cancer cells is epithelial mesenchymal transition. This is a process by which cells lose their epithelial properties and gain mesenchymal properties. This normally occurs during the development process in embryos, allowing cells to migrate to their appropriate destinations in the embryo. It also occurs in wound healing, tissue regeneration, and fibrotic reactions. But in all of these processes, cells stop proliferating when the process is complete. Malignant cells that metastasize often undergo EMT as an important step in that process, but retain the capacity for unregulated proliferation. Malignant cells that gain access to the circulation must then repeat those steps at a remote site, find a hospitable niche in a foreign tissue, avoid detection and elimination by host defenses, including the immune system, and induce the growth of new blood vessels. The rate limiting step for metastasis is the ability for tumor cells to survive and expand in the novel microenvironment of the metastatic site, and multiple host tumor interactions determine the ultimate survival. Normal tissues have stem cells capable of self renewal and repairing damaged tissue, whereas the majority of cells in normal tissues do not have this capacity. Similarly, only a small proportion of the cells within a tumor are capable of initiating colonies in vitro or forming tumors at high efficiency. For example, AML and CML, that is acute myeloid leukemia and chronic myeloid leukemia, have a small population of cells, estimated to be less than 1%, that have properties of stem cells, such as unlimited self-renewal and the capacity to cause leukemia. These cells have an undifferentiated phenotype, Thi1 CD34 positive, CD38 negative, and do not express other differentiation markers, and resemble normal stem cells in many ways, but are no longer under homeostatic control. Solid tumors may also contain a population of stem cells. It is not yet known how often cancers may originate within a stem cell population. Cancer stem cells, like their normal counterparts, have unlimited proliferative capacity and paradoxically traverse the cell cycle at a slow rate. Cancer growth occurs largely due to expansion of the stem cell pool, the unregulated proliferation of an amplifying population, and failure of apoptosis pathways. Implicit in the cancer stem cell hypothesis is the idea that failure to cure most human cancers is due to the fact that current therapeutic agents do not kill the stem cells. Identification and isolation of cancer stem cells will allow determination of the aberrant signaling pathways that distinguish these cells from normal tissue stem cells. These would serve as potential therapeutic targets. Plasticity and resistance. Plasticity. This means the capacity of cancer cells to shift dynamically between a differentiated state with limited tumorigenic potential and an undifferentiated state or cancer stem-like cell state, which is responsible for long-term tumor growth. Cancer cells, and especially stem cells, have the capacity for significant plasticity, allowing them to alter multiple aspects of cell biology in response to external factors, example, chemotherapy, radiation therapy, inflammation, immune responses, etc. A major problem in cancer therapy is that malignancies have a wide spectrum of mechanisms for both initial and adaptive resistance to treatments. Thus, most metastatic cancers eventually become resistant to the therapy being utilized. Overcoming resistance is a major area of research. This figure shows Warburg effect. That is, the normal cells concentrate their metabolism more on oxidative phosphorylation to have more high energy phosphate bonds in the form of ATP. Cancer cells get their energy by aerobic glycolysis to get more components for the building blocks of their various organelles. Cancer metabolism. One of the distinguishing characteristics of cancer cells is that they have altered metabolism as compared with normal cells in supporting survival, their high rates of proliferation, and ability to metastasize. Complicating studies evaluating metabolic differences between normal and malignant cells is that there is heterogeneity in metabolism between different cells within a cancer. Malignant cells must focus a significant fraction of their energy resources into 1. Synthesis of proteins and other molecules, that is, building blocks required for the production of new cells, while 2. Still maintaining sufficient ATP production to survive and grow. Many cancer cells utilize aerobic glycolysis, that is the Warburg effect. You see the figure shown earlier, to metabolize glucose, leading to increased lactic acid production. Normal cells utilize oxidative phosphorylation in mitochondria under aerobic conditions, a much more efficient process for generating ATP for energy utilization, but one that does not produce the same level of building blocks needed for new cells. One consequence is increased glucose uptake and utilization by cancer cells. The inefficient utilization of glucose by malignant cells also leads to a need for alternative metabolic pathways for other compounds as well, one of which is glutamine. Similar to glucose, this provides both a source for structural molecules as well as energy production. Similarly to glucose, glutamine is also inefficiently utilized by cancer cells. Cancer cells can also take up nutrients released by surrounding cells and tissues, increasing the complexity of successfully therapeutically inhibiting metabolism in cancer. See the figure Warburg effect versus oxidative phosphorylation again. In most normal tissues, the vast majority of cells are differentiated and dedicated to a particular function within the organ in which they reside. The metabolic needs are mainly for energy and not for building blocks for new cells. In these tissues, ATP is generated by oxidative phosphorylation that efficiently generates about 36 molecules of ATP for each molecule of glucose metabolized. By contrast, proliferative tumor tissues, especially in the setting of hypoxia, a typical condition within tumors, use aerobic glycolysis to generate energy for cell survival and generation of building blocks for new cells. Mutation in genes involved in the metabolic process occur in a number of cancers. Tumor microenvironment, angiogenesis, and immune evasion. Tumors consist not only of malignant cells but also of a complex microenvironment including many other types of cells, including lymphocytes, macrophages, myeloid cells, other inflammatory cells, fibroblasts, and fat cells. Extracellular matrix, secreted factors like growth factors and hormones, reactive oxygen and nitrogen species, mechanical factors, blood vessels, and lymphatics. This microenvironment is not static, but rather is dynamic and continually evolving. Both the complexity and dynamic nature of the microenvironment enhance the difficulty of treating tumors. The microenvironment can contribute to resistance to anti-cancer therapies through a number of mechanisms. Obesity and cancer. Significant evidence links obesity and the increased risk of developing certain cancers, including postmenopausal breast, colorectal, ovarian, endometrial, esophageal, gallbladder, thyroid, and kidney cancers, among others. Less certain are the mechanisms responsible for this risk. As outlined above, cancers arise in an environment with multiple factors, many of which can stimulate cell proliferation. Obesity impacts a variety of factors including hormonal factors, altered metabolism, especially adipose metabolism, and mediators of inflammatory response that all can impact the development of malignancy. Obesity is associated with a number of hormonal changes, including high insulin, glucagon, and leptin levels that can stimulate growth of cells. It also leads to insulin resistance, which may contribute to cancer cell development in part by increasing insulin-like growth factor 1, that is IGF-1 levels. Obesity contributes to an inflammatory environment in a variety of ways, including increased levels of inflammatory proteins such as IL-6 and TNF-alpha. Obesity is associated with decreased survival, likely due, at least in part, to the impact of obesity on hormonal factors in development of certain breast cancers. Mechanisms of tumor vessel formation. One of the critical elements of tumor cell proliferation is delivery of oxygen, nutrients, and circulating factors important for growth and survival. Thus, a critical element in growth of primary tumors and formation of metastatic sites is the angiogenic switch, that is, the ability of the tumor to promote the formation of new blood vessels, including the recruitment of vascular endothelial cells, ECs. The angiogenic switch is a phase in tumor development when the dynamic balance of pro and anti-angiogenic factors is tipped in favor of vessel formation by the effects of the tumor on its immediate environment. Angiogenesis consists of several steps, including the stimulation of ECs by growth factors, degradation of the ECM by proteases, proliferation and migration of ECs into the tumor, and the eventual formation of new capillary tubes. Tumor blood vessels are not normal. They have chaotic architecture and blood flow. Tumor vessels are tortuous and dilated with an uneven diameter, excessive branching, and shunting. Tumor blood flow is variable with areas of hypoxemia and acidosis leading to the selection of variants of tumor cells that are resistant to hypoxemia-induced apoptosis, often involving the loss of p53 expression. Tumor vessel walls have numerous openings, widened interendothelial junctions, and discontinuous or absent basement membrane. This contributes to the high permeability of these vessels, and together with lack of functional intratumoral lymphatics causes increased interstitial pressure within the tumor, which also interferes with the delivery of therapeutics to the tumor. Tumor blood vessels have a deficit of perivascular cells such as pericytes and smooth muscle cells that normally regulate flow in response to tissue metabolic needs. Unlike normal blood vessels, the vascular lining of tumor vessels is not a homogeneous layer of ECs but often consists of a mosaic of ECs and tumor cells. These cancer cell-derived vascular channels, which may be lined by ECM, secreted by the tumor cells, are referred to as vascular mimicry. Lymphatic vessels also exist within tumors. Evasion of the immune system by cancers. The immune system plays a critical role in maintaining organismal integrity, including defense against pathogens, as well as preventing and limiting the growth of cancers. There is a complex interaction between cancer and the host from the development of the first malignant cell to the establishment of a clinical cancer and its subsequent growth, invasion, and metastasis. The immune system plays a critical role in the prevention of cancer development. This is exemplified by the increased risk for cancer development in individuals who are significantly immunosuppressed, such as by inherited defects in mechanisms important for immune function, the immunosuppression necessary to maintain organ transplants, and immunosuppression seen from certain infections such as human immunodeficiency virus. There are two components of the immune system. The first is innate immunity present in the organism. not dependent on prior exposure to a specific antigen, such as a pathogen or malignant cell. It tends to be general and not specific. The second is the adaptive immune component, which depends on the innate immune component for activation and provides the specificity to the response with significant expansion of cells to target the specific antigens present on the pathogen or malignant cell. Thus, while the innate process provides the first line of defense, the adaptive process is necessary for the specificity of response and providing memory to more rapidly attack cells should the pathogen infection recur or the malignant cells grow. The immune system has to be tightly regulated to allow for clearance of unwanted antigens while preventing an immune-mediated attack on the cell. Not surprisingly, since cancers arise from normal cells within the body that have a variety of processes to prevent destruction by the immune system, they have a variety of mechanisms that allow them to evade detection and elimination by the immune system. These include 1. Down regulation of cell surface proteins involved in immune recognition, including MHC proteins and tumor-specific antigens. 2. Expression of cell surface proteins that inhibit immune function, including members of the B7 family of proteins such as PDL1. 3. Secretion of immunosuppressive proteins. 4. Recruitment and expansion of immunosuppressive cells such as regulatory T cells, which are important for maintaining tolerance against self-antigens. 5. Induction of T cell tolerance. And 6. Downregulation of death receptors. Due to the marked heterogeneity of cells within a cancer, a variety of immune suppressive mechanisms are continuously occurring and changing. Summary: better understanding of the biology of early cancer development and technological development to improve sensitivity and specificity in detecting cancer-specific molecules, example, mutated genes, provide hope that approaches for earlier detection of cancer cells can be developed. So, dear viewers, this is the end of part 4 in the 4-part series on cancer cell biology in the Medicine for Anyone in the YouTube channel. Thank you for watching the video.